anticipating them asking for a seat change, but they did not. They did not do that. But if you came to this talk either excited or afraid to see pictures of mini parasites, you are going to be disappointed either way, because I did not include a lot of pictures of parasites. I do have some cartoon images. I knew I would be speaking before lunch, and I thought it would be maybe more appropriate to make it to where you don't leave this talk hating the idea of animals that live inside of other animals, to avoid having some of those images permanently just attached to your brain. So this talk it's really two talks in one. The first half is a confession of how I just got everything completely wrong in my idea of these animals that live inside of other animals. And really, it's not me alone. I think it's parasitologists really across the board have got these really, really wrong. And then the second part of it is how can we actually get this right? And giving you some specific examples, some take-home of specific species and how we can get them right. And so the outline for my talk, if I can get this to work, even if I can't get this to work. Oh, I see what, it does like to skip ahead. You see here, there's a bit of a lag. Okay, so I also, I can't see what's coming next, so sometimes it'll surprise me as much as it surprises you, which will always be good. So the outline of my talk, the first 2 points are really that first half, that confession of how I got things just completely wrong. And the first one, I think I could be forgiven for it, if not by you, at least by myself. But the second one, I don't think that there's any reason why I should have gotten the second one wrong. But first one is how I was wrong about symbiosis. Second one, how I was wrong about parasites. And then specific symbionts we all were wrong about and how we can get them right. And then a little bit of how my view has changed to an opposite extreme with an example of some research that I am currently doing. So some definitions to get us started. Symbiosis, I'm going to define this, and when I use this term, as an interaction between two species living in close proximity. And the key there is really the two separate species when we're talking about symbiosis. So we're not talking about cooperation among two individuals of the same species. We're really talking about, like what Dr. Blaschke illustrated, a caterpillar and an ant. Two completely different species living in really close proximity. And something that might be added to this, should be added to it, is for a duration of time, living in close proximity for a duration of time. Because a predator and its prey live in close proximity for a very short period of time, and then that relationship ends. But we're talking about something that's, it's a little bit more involved, and it's a little bit more long-lasting. And then another term that I'm going to use often, and it's a clunky term. It works really well biologically. It's not as easy to say, which is why I think part of the reason why we still use the term parasite, even though we can all agree that it's not actually a parasite. And Dr. Blaschke did a good job, using a parasite as an illustration of a despicable part of God's creation. And those that are truly parasites really are. They are despicable. And I think we either need to work towards their eradication or work towards redeeming them, figuring out what they were designed and created to do and eliminate them from where they were not designed to be there. But an endosymbiont, so this is it's one of the members of this symbiosis, one of the members of the symbiotic relationship, but it lives inside the other organism. So I will, I think, once use the term ectosymbiont. But for the most part, I'm using the term endosymbiont. So an animal, and I focus on animals. There are non-animal endosymbionts as well, but they are beyond my ability to think and understand. And so I focus on animals that are living inside of another animal. And then that gives us the term for the other animal in this relationship, in the symbiotic relationship, the host. So this is the one that is the house for its symbiotic partner. And again, we're talking about living inside, and this is two different species, and there's some duration to this. And I think Dr. McLean asked Dr. Blaschke at the beginning the difference between a parasitoid and a parasite. And parasitoids mostly insects. I think there are some parasitoids outside of that group, but they killed their host. They may live inside it for a very short period of time, but endosymbionts and even parasites are really, really good at keeping their hosts alive because their survival depends upon it. All right, so here was my idea. This is how I got it completely wrong. This is my confession. I had this idea that symbionts are very rare, especially endosymbionts. Endosymbionts are incredibly rare. Endosymbionts are unimportant, and this is in the terms of both to the health of most organisms and to the ecosystem, that they are just unimportant and can be ignored. And the last one is that symbionts, and especially endosymbionts, were all created free living and then have developed this kind of symbiotic lifestyle since the creation event. And these are all wrong. So as you write these down in the notes, it's kind of fun to write things down that I'm telling you are wrong. But you can share, my experience where I just got things completely messed up. And this was one I think I can be forgiven for because I didn't actually see a parasite live and in person until I was 23 years old. And I was spared from much of it growing up. I grew up here. I went here as an undergraduate, and I didn't see a parasite for the first time until I joined the faculty at Cedarville, where Dr. McLean was a student, and had a colleague that encouraged me to do something different with the invertebrate zoology class that I was assigned to teach. He said it's not a very popular class, because a lot of the organisms that you cover are already covered in general zoology. And so he said, you should do something different with it. Why don't you make it a parasitology class? I said, that sounds wonderful. And he said, you should start with raccoons. And little did I know at the time that raccoons are basically a lunchbox for all kinds of animals. You open them up and there's just all kinds of treasures inside. Maybe a lunchbox isn't the best analogy. Maybe it's like a trash can. That works actually as a nice analogy for the trash panda itself. But he said you should start with raccoons because they're just so easy to come by. There are more raccoons in the state of Ohio than there are people. And I had a 10-minute drive to work and would drive by at least one new raccoon every day. At least one. Some days more. And so we started there, and as we started working through these animals and opening them up, they're just full of all kinds of things that lived inside of them. And so when I came across this for the first time, my ideas started to shift and away from some of these things. And what really got my ideas to shift was research that was being done by parasitologists as I started to get into this field of parasitology, realizing that endosymbiosis is incredibly common. It's not rare at all. In fact, nearly half of all animal species live in some kind of a symbiotic relationship with another animal, a long-term relationship. About 20% of those are endosymbionts. They live inside of other animals, at least for part of their life cycle. And half of the 35 animal phyla have at least one group of endosymbiotic species. And so as I'm reading these things, my ideas are shifting away from these things are incredibly rare to these things are actually really common. And then so that last point where I think that God had created these as free living, I'm starting to get very nervous about this idea of how common this is and how unlikely it is to actually develop some of those. So the next one of that they are relatively unimportant. This is the Allegheny wood rat, and it has been extirpated from almost its entire range by the parasite that I studied for my PhD, raccoon roundworm. And I'll talk more about raccoon roundworm as one of those things that we got wrong for the most part when it's where it's supposed to be. But we got it very right when it ends up in places where it is not supposed to be. And so these endosymbionts are not unimportant, and I'll talk about this more in my second talk of what did God actually design these to do initially, and how do we get back to that, or how do we protect that while eliminating some of the things that God did not design them to do. But this is a good example of the ecological importance of parasites. And the unfortunate thing about parasitology, and I share these things that bother me, so that you can be bothered by them too, maybe. is the only parasites we really know a lot about are those that either cause human health issues or are economic or cause issues in economically valuable animals. And so there are a lot of parasites we just don't really know very much about because they are doing things like extirpating the Allegheny wood rat from its range. And I don't know how many people there are that are really sad about that. Hopefully after the first talk, you're a little bit sad about that because, this is one of those things that adds to the diversity of God's creation. So what really did this for me of changing my views on that last point, that these were created to be free living, are these things. Well, one, endosymbionts, when you compare them to free living forms, they're much simpler. They've either moved into this lifestyle and lost a lot of their diversity, of their anatomical diversity, or they were created basically without the ability to have a free living stage. Many of these organisms completely lack all sense organs, which makes sense. If you spend your entire life living inside of another animal, there's not a lot of reason to be able to see or to detect chemicals other than those that are inside of your host. Also, endosymbionts have increased reproductive output, and this is something it's more complicated than just saying, well, if I'm not investing resources in this, I can invest resources in this. Many of them, like tapeworms included, which I did not include any pictures of, even cartoons of tapeworms, but they are basically just bags of reproductive organs. I mean, their body plan is extraordinarily different than anything we have in a free living animal. And then another thing is endosymbionts often invade host tissues. So 4 years ago, I believe it was, Dr. Blaschke presented a paper at the Creation Biology Society, which was the meeting where we met a couple years before this, talking about the difficulty of getting the diversity of parasite forms that we have in a very short amount of time since the creation. And one of the things that he talked about in this paper was that we have this group of organisms that are called facultative endosymbionts, or facultative parasites, that can be free living, but they don't mind actually shifting their diet to eat materials that are inside of a host. But the thing is, when you start invading host tissues, now everything's different. It's completely different because now you have an environment where the chemical environment is like nothing you find in a free living environment. And even to controlling water, the movement of water across the cell is extremely difficult when you are built to live in fresh water and then you end up inside of the tissues of another animal. And then this last one, and Dr. Blaschke talked a little bit about this, and I will talk about this more during this talk and in the one after lunch, is these endosymbionts regulate the immune system. And they do so really, really well, really, really well. And I've got some figures, and I'll talk to you about the different divisions of the immune system and what they actually do to give you something that Dr. Francis would do if he were here, but he is not, so I will do my best to get you what you need. And so this is, I got this completely wrong, and this is what's actually true. Symbionts are common, extremely so. In fact, some parasitologists suggest that there's at least one nematode worm for every species on Earth. Every animal species on Earth has its own nematode worm to call its own, to take care of it, to care for it, to feed it, willingly or unwillingly. And so symbionts are extremely important, ecologically and otherwise, and they were created to live inside of other organisms. I don't think there's any other explanation for what they have other than that God created them to live where they are. So that was the first thing I got wrong, and again, I will forgive myself for it because I just had not ever experienced endosymbionts before. But the second one I should not have got wrong. How I was wrong about parasites. So now I'm believing endosymbiosis is incredibly common, and we're working on these raccoons, and I start to see everything, not just raccoons, but every animal, as just this lunchbox or this trash can. It's a great analogy, full of other things, and it starts to terrify me. I start having nightmares. Part of it was because I would put some of these animals in my freezer at home to wait until I could take them to school. But I started just seeing everything as just this something so wrong about this world that God's made. And I started seeing everybody's illness as the result of a parasite. So somebody's got a headache, and I'm like, man, you probably have some kind of brain-eating amoeba. Should probably start calling people now because you've got maybe 20, 30 minutes tops. And it just shifts me over to something that I don't think there's a real excuse for, because it's just so obvious that that's not what God allows with his creation. And so before I share with you what changed my ideas on here, we've got some more definitions to put out. So I'm going to define a parasite as a symbiont that harms its host. So it harms its host in some way. A commensal is a symbiont that neither harms the host nor helps it. It lives there. It's with its symbiont. And then a mutualist is a symbiont that benefits its host. And this is what Dr. Blaschke showed with the caterpillar and the ant. Okay, a real nice mutualistic relationship between them. So my idea at this point is all endosymbionts are parasites and need to be eradicated. And Dr. McLean might actually be able to attest to this if he remembers the class that he took with me and hasn't blocked all of those memories from his mind. But I would, every time we got to a parasitic group of animals in this general zoology class, I would end it with, they need to be eradicated. There's nothing good here. There's nothing left that is good from what God designed. And then I start just thinking about God's creation, and it's crazy. And I think I was so obsessed with the grotesqueness of these animals that I could not get past it. And then this is one that really helped me. So I don't know if you know this animal, but this is a blobfish. And this quite possibly might be the ugliest thing I've ever seen in my life. And I've seen lots of parasitic worms, both in situ and out. But God loves these things, right? And we can't just look at this thing and say it's ugly. Now, some people suggest that actually where it lives under the high pressures, it might not be this ugly. It might actually be more rounded out. And even so, it's still, it's not the most beautiful thing. But God loves it. God loves what he made, and God enjoys what he made. And so these things that are grotesque shouldn't just overwhelm my thinking on these things. And then as I start reading the account of God's promise to Noah, I was always taught that the covenant God made with Noah in Genesis 9 was made to Noah, but it's actually made with all of creation, which is really interesting as you read through Genesis 9, and God promises to never destroy the world by flood again. It's very clear over and over and over again that this is a promise being made to all of creation. And so I'm sitting there thinking about this, and I'm like, okay, so God loves the things even that are ugly, and God loves his creation so much. Like he promises not to destroy it by the flood again, and actually to intervene to keep things from getting as bad as it got before the flood. And then I start to do things like this. So Answers in Genesis invited me to write a paper for the magazine on blood-feeding animals that feed on blood, and the incredible design that it requires to feed on blood. So blood is an incredibly difficult food source because of the iron content in blood. And so if you get a nosebleed and you happen to be unfortunate enough to consume some of that, you almost immediately get an incredible stomach ache and stomach distress, because digesting iron is very difficult. And these animals that are feeding on host tissues have some just incredible adaptations to be able to do this. And so I'm not saying that God designed these animals to feed on blood, because I don't think that they did, at least not all of them. Some of them that lived inside of animals I think could have been designed to feed on blood. But even these endosymbionts have just incredible design elements. And then what really did it for me is studying other pathogens. So we have all these disease-causing organisms, bacteria, viruses, fungi. And the overwhelming majority of these species are not pathogenic. The overwhelming majority of bacteria, even those that live inside and on us, are at worst commensal and neither harm nor benefit us. And many of them benefit us, right? Where you talk about rebuilding your gut health so that you can digest food properly. And the same thing is true of viruses. The overwhelming majority of viruses are just living out their existence, although they're not really living, carrying out their existence and are non-pathogenic. And then most of the issues actually associated with parasites themselves are actually our immune system overreacting to their presence. And so now I'm like, okay, I was wrong about endosymbiosis and I was wrong about parasitism. And so the overwhelming majority of these things are endosymbionts, probably commensal and maybe even mutualistic. And so now that's the first part of the talk, the confessions of where I was wrong. And now I want to leave you with some specific species that we can do better about getting right. And so the first one, Ascaris lumbricoides, which is the human intestinal roundworm, and then Trichuris trichiura, which is called the whipworm. Nicator americanus is one of the hookworms that Dr. Blaschke was talking about during his talk. And then this one at the end, I affectionately refer to as my parasite, not because I have it. In fact, I'm almost convinced I do not, because that's the one that I studied for my PhD, Balis ascaris procyanus, which is the raccoon roundworm. And so we'll start with Ascaris lumbricoides. And all of these, by the way, I think every parasitologist would agree that these 4 species are not parasites when they are in their normal host. They are not parasites. They are not harming. At worst, they are commensal. And yet we still refer to them as parasites because endosymbiont is long and clunky. And so Ascaris lumbricoides is an interesting one because as old as records as we have of human writing, we have references to this parasite. And we have found eggs of this parasite in mummified remains in ancient Egypt. This is one that, this is an endosymbiont that, see, I called it a parasite, and I shouldn't have. This is an endosymbiont that's been with us for a long time, and it's a really, really interesting one. So you have to swallow the eggs, so you have to touch a surface that's contaminated with these, which isn't all that hard because I think the current estimates are about 1/4 of the world's human population has this endosymbiont living inside of them. So you have to touch a surface contaminated with the eggs, and then the eggs hatch in your small intestine, and then the worms travel. So they move from your small intestine, and they move all the way through your body to get through stages of development to end up back in the intestines as adults. And they can live as adults for 60 years in your intestines, releasing 100,000 to 300,000 eggs a day, and just being there and not causing you any harm whatsoever. In fact, at the worst, they're commensals unless you have a really, really large and a large number of them, or you swallow a number of eggs at the same time, and we're talking about maybe tens of thousands of eggs at the same time. But they actually are even beneficial. This is one of the parasites, in addition to hookworms, one of the endosymbionts that is being used to treat autoimmune issues. And right now, they're working on just pulling the antigen, the proteins on the surface of the worm, and turning it into a pharmaceutical drug that you can take rather than infecting yourself with the endosymbiont intentionally. Yeah, although the videos that you get when you swallow a camera are a lot more interesting when you actually have the worms living inside you than when you're just using the actual pills. So, and then this one's whipworm, Trichuris trichiura, and it's fairly similar to Ascaris lumbricoides in that it lives in the intestines, but it doesn't do that really complex, circuitous route through the body that the Ascaris lumbricoides does. It ends up in the intestines and it stays there. But this one is really interesting in that it burrows into the wall of the intestine. And so this is one that there's no free living stage for this. There are no free living, I don't think, ancestors of this. This was designed to live inside of humans, that this was actually part of God's design to have these animals living inside of us. And they, again, are there, and they are not causing any harm. And in fact, actually, like Ascaris lumbricoides, ensuring that your immune system behaves appropriately. And Dr. Francis, I don't know if he's going to talk about this in his talk, but he talks about that the design of the immune system was likely to keep our gut bacteria in check, That it was a mechanism to make sure our gut bacteria stays in the right proportions and the right amounts and stays where they're supposed to be so that we have optimum health. And the interesting thing about these animals is they ensure that our immune system behaves appropriately and that it actually functions the way God designed it to function, and that is likely to regulate our bacterial communities. All right, and so here is the hookworm. I chose Nicator americanus, but there's also other species of hookworms. Now this one has a free living stage. So they've got a couple of larval stages that live outside of a host. And so in order to get into the host, they will actually stand up in their larval stage and wave back and forth like a blade of grass, but they're tiny. You can't see them at this stage. They're microscopic, and wait until you step on them barefoot, and then we'll burrow through your feet and take a route similar to Ascaris lumbricoides to end up ultimately in your intestines. And these endosymbionts feed on blood. And I think again, talking about how difficult it is to digest blood, these seem like they were designed to feed that way. And the interesting thing about these do the same thing that Ascaris lumbricoides and Trichuris trichiura do, in that they ensure that your immune system behaves properly. But it takes far fewer of them to actually make that work. So for it to work for Ascaris lumbricoides or Trichuris trichiura, you need several thousand of these worms living inside of your intestines to make your immune system behave appropriately. With these, you need maybe a couple of 100. And so I had a colleague at Biola who had intense Crohn's disease, and he ordered helminths from Europe and took them and gained remission from them. And they always started him on a regimen of hookworms and then shifted it over to whipworms afterwards. And then the last one, again, the one I affectionately refer to as my parasite, Balis ascaris procyanus, the raccoon roundworm. This one is probably, in terms of human health, one of the deadliest parasites on Earth. When it's in humans, it is most certainly a parasite. It does not belong there. And this is the one that I talked about its ecological importance in the Allegheny wood rat, and has almost eradicated that animal from its native range. But when it's in the raccoons, it behaves like Ascaris lumbricoides does in us. It regulates the raccoon immune system, making sure it behaves appropriately and the raccoons don't overreact to other things. Raccoons get autoimmune diseases like we do as well. It's probably because they eat our garbage and whatever's in there, but raccoons suffer from many of the things that we do. But these worms ensure that their immune systems behave properly. And these worms actually do something really interesting in the raccoons. So there's a thing that endosymbionts do that's called parasite-induced trophic transmission. And it's a fancy way of saying that parasites, endosymbionts, parasites specifically, are really good at getting themselves to their next developmental stage. And so many of these symbionts require multiple hosts in order to complete their life cycle. And so what they'll do is they'll change the behavior of an intermediate host to bring themselves into contact with the next host. I think the best example of that is a parasite called Leucochloridium paradoxum, which I wanted to put a picture of this, but it's a little bit disturbing. What they'll do is they'll climb into the eye stalks of snails, and they'll create like a strobulation. Strobulation, does that work? It's like a strobe effect. And so it makes the eye stalk of the snail actually look like a caterpillar. And it causes the snail to climb up into trees where the caterpillars typically live. And then birds eat the snails thinking that they're a caterpillar, and the birds are the final host for this parasite. But what my group published about this parasite, Balis ascaris procyanus, is it actually is the first documented case of parasite-induced trophic transmission in the final host. And so what it does is it changes the raccoon's behavior to make the raccoon eat foods that are more attractive to the intermediate hosts of this endosymbiont. So it makes the raccoons eat foods that make it more likely that the larvae of these endosymbionts will end up in a place where they can develop. And it's really fascinating that we have a worm in raccoons that does what Ascaris lumbricoides does for us, but it also actually shifts the raccoon behavior a little bit, which is really fascinating to me. And so those are the four endosymbionts that I think my hope is that you can take away with that these things are incredibly well designed. They're what you might call the capital B beautiful, even though they are grotesque, because they represent the nature of God, this diversity within these organisms, and the unity of that as they work together and labor together to make organisms work the way they were supposed to work. And so now my view of these is just completely different. And so I'm no longer sitting there looking at people and diagnosing every ailment they have as the result of a parasite, but I'm actually encouraging people to actually treat their ailments with parasites, you know? And not only that, but looking at these to try to solve other ecological problems. And so one of the research projects we're working on now is to try to limit predation on ground nesting bird nests. And so here is the nest of an ovenbird. Ovenbirds are ground nesting birds. And ground nesting birds have a number of issues associated with having their nests on the ground, one of which is they often fall prey to a number of different predators. And so one of the things we're working on is to try to figure out a way to limit predation on these nests. And so one of the things we noted is that some of the nests that survived and actually had the chicks fledge were just the nesting material was almost saturated with these ectosymbionts. So these animals that will live on the outside of the hatchlings and will feed on them. And there are a number of people that have done some work to show that there's no net loss of fitness for the birds when they are being fed on by these ectosymbionts. So they are commensals. Even though they are taking resources away from the hatchlings, they aren't taking enough away to actually result in the hatchling success suffering. And so what we predicted with some of these nests that were successful being almost saturated with these ectosymbionts is that the predators are deliberately avoiding feeding on nests that have these ectosymbionts, because while they might not cause very many issues for the birds, they might very well for the predators. And so we predicted that if we were to put nests out that we deliberately contaminated with these ectosymbionts, that we could drop the level of predation on these nests. And it worked, actually. So we made some artificial nests. Jack was actually part of this, right? The initial stages. And so we made some nests, and the nests that were saturated with ectosymbionts, even nests that didn't have that many, were preyed on significantly less than the other nests. That the predators were deliberately avoiding nests with these ectosymbionts in the material. And what's interesting here is if these ectosymbionts were actually designed to be part of this bird's life history, there's something built into that design that actually protects these birds from falling prey to predators, which I think is really fascinating, and it is really important for us as we start to build models of how do you actually keep animal death from happening. How do you actually make that happen? What changed in creation to actually shift it over to where predation is one of the dominant, feeding strategies within God's creation? Because I don't think that was designed, but was a result of the fall. So that's it for this, except for to leave you with a question that we will attempt to answer in my second talk, and that's we have this entire category of organisms that we'll collectively call endosymbionts, and to see what did God design these to do? What was their intent? We have things like seed-eating birds, that it's very clear what they were created to do. They were created to help disperse plant species, or fruit-eating animals, that they were created to help disperse the seeds. And we have this entire group of organisms, And I think we have a really important question to ask and to answer as we attempt to make some of these models for what creation was like before the fall. And how do we, as we adopt it and as we try to get things back to a creation that more properly and effectively represents the diversity and unity within God, how do we get there? And the nice thing about this is, I'll talk about this more after lunch, is most of these endosymbionts are not parasitic. And so we actually have some of these that are right now doing what I think God designed them to do. So